Today topic is Embryosac and its types in Angiosperm plant or flowering plants. Before we start about Embryosac and its type in Angiosperm, let us know briefly about female reproductive parts in flowering. Female reproductive part is pistil, are also called carpels, are also called gynoecium, or also we can use another term, that is mega. Here, based on the number of carpels, these carpels are divided into two types. If only one carpel is present, such carpels are called mono. If more than two carpels are present, such carpels are called multicarpellary condition. This monocarpellary generally present in hibiscus plant. Here the multicarpellary, if this multicarpellary in which if more than two carpels are free from each other. They are free from each other. Such carpels are called apocarpal. If more than two carpels are fused or united together, such carpels are called syn. These apocarpus are generally seen in a plant called Michaelis plant. And these syn carpus conditions are generally seen in papaya. So this is female reproductive parts, that is pistil or carpel. And now we move on important, that is carpel structures. Here each carpel or pistil made up of with three parts. They are here. The first part stigma, second one is style, and third most important part, that is ovary. Like this, each carpel like this is a each carpel or pistil. And here we have this part is called stigma, this region is called style, and this region is called ovary. Now we'll move on in detail of ovary. Generally, this ovary has cavities or empty space within ovary. And this empty space of the ovary is called ovarian lacule, is a empty space in ovary. And this ovarian lacule contain a structure or a tissue called placenta. And from this placenta, there is a structure which is arise from this placenta. The structure which is arise, so the structure is called ovule. And this ovule also called megasporangium. So like see, this is the empty space in the ovary called ovarian lacule, in which we have contained like this structure called placenta. Like this placenta we have. From this placenta, there are structures which is arise like this. Now this arise structure from placenta is called ovule, also called megasporangium. Now we will discuss in detail structure of ovule or megasporangium. Here structure of ovule are also called megasporangium. Common type of ovule present in angiosperm, that is anatropous ovule, is a common type of ovule present in angiosperm plant. This is a common type of ovule. Generally, we have different types of ovule. We will discuss in another video lecture. But in angiosperm, most common type of ovule is anatropous ovule. And this, now let us discuss the in detail structure of ovule. Here, this is the placenta, and from the placenta, there is a stalk which is arise like this. And this stalk of the ovule, it is a stalk of the ovule, is called funicule. And this funicule here, this the structure which is arise from, that is called placenta. This is the placenta. From placenta only the ovule will arise. And this funicule is a stalk of the ovule which arise from the first placenta. And these funicule which mainly helps in connect placenta and body of the ovule. We will discuss later. Which mainly helps to connect placenta to body of the ovule. And from this stalk there is a body of ovule which is arise like this. Yeah. So this is what body of the ovule. See here, the stalk of the ovule, that is called funicule, which connect placenta and body of the ovule. And the region where the body of the ovule which attach to the funicule, here this region, and here we have one hilum, a structure called hilum. So hilum is a junction between ovule and funicule. Hilum is a junction between body of ovule and funicule. Now we move on body of ovule. Generally, this body of ovule, which is contain a protective sheath around it, like this one, or it may be sometime two. These two are protective layer, protective tissues, and these protective tissues are called in generally mostly there are bitegument, means there are two integuments are present. Rarely unitegument, and if there are two integuments means one integument is outer integument, another one is inner integuments, like this. See, this is the outer integument and this one inner integument. and it is a protective tissues which is present around the body of ovule. And these protective tissues which mainly protect these tissues which is present inside the ovule, that is nucellus. Here we have nucellus tissues, very important tissues. Yeah, here these nucellus tissues is a mass of parenchymatous cells, which is enclosed within the and one more important point, and these nucellus which contain reserve food material, or which store reserve food material. And these nucellus cells are made up of thousands of cells. Generally in nucellus there are thousands of cells are present in nucellus, and among these thousands of cells, any one of the cells will undergo changes or modification to become megaspore mother cell. MMC means here megaspore. Sorry, yeah, here MMC means megaspore mother cell. And these megaspore mother cells, which is form only any one of the thousands of nucellus cells, and these megaspore mother cells, yeah, here these megaspore mother cells which undergoes first meiosis division, then it follow mitosis, several mitosis division to form embryo sac. And here the embryo sac is also known as female gametophyte. Here the female gametophyte are embryo sacs which is formed from megaspore mother cells by undergoes meiosis division, then it followed the various or several mitosis divisions. So likewise the embryo sac will be formed from the female reproductive parts. Now we'll here, and these embryo sacs which is present here, and the formed embryo sacs which is present here. For example, see, this is how embryo sac will be formed. And these embryo sac here, yeah, here I'm writing. So these formed embryo sacs has two ends. The upper end here we have called chalaja end, like here chalaja end. Here also we can use chalaja end. And here this side we have micro. It has two ends. This ovule contain two ends, or also we can use the term embryo sacs also contain two ends. One is chalaja end, another one is micropylar end. For example, here upper side of the embryo sac has chalaja end, and lower side here we have micropylar. And this is what embryo sacs. So this embryo sacs only this one. And these embryo sacs which contain three different types of cells. For example, here towards the micropylar end, in center we have egg cells. See, this is what egg cells are also called ovum. And besides egg cells, we have two important cells. They are called synergids. These two synergids and one egg cells together called egg apparatus. And at center we have central cell. It contains two nucleus. Those two nucleus are called pro, and it is a two-end stage. And here at the chalaja end we have three cells. They are called anti cells. Likewise, embryo sacs contain three different types of cells. At chalaja end we have antipodal cells. At central we have central cell which contain two pronuclei. Towards micropylar end we have egg apparatus, and this egg apparatus contain one egg cells and two synergid cells. So this is the structure of embryo sac. Now likewise, and it is a complete structure of ovule, and inside the ovule we have embryo sac, and inside the embryo sac we have egg. Now we discuss types of embryo sac. Yeah, types of embryo sac in angiosperm. Here, on the basis of functional megaspore. On the basis of functional megaspore, we have three types of embryo sac. There, first one is mono type of embryo sac. Second one is bisporic type of embryo sac, and third tetrasporic type of embryo sac. So all these types based on only functional mega. How means in monosporic type of megaspore, in which only from one functional megaspore only embryo sac will be developed, or embryo sac will be formed. Such type of embryo sac is called monosporic. And in bisporic, from two functional megaspore only, from two functional megaspore complete embryo sac will be formed. And in tetrasporic, here from all four functional megaspore, this embryo sac will be formed. So based on this functional megaspore only, we have three different types of embryo sac. Now we will discuss each type in detail. Yeah. Here first monosporic type of embryo sac. So here this monosporic type of embryo sac formed from unfunctional mega. Note, very important note here, in which monosporic type of embryo sac, in which one meiosis and three mitosis occur. But in Enothera type of monosporic type of embryo sac, we will discuss generally monosporic type of embryo sacs will form by two methods. In that, one is polygonum type of monosporic type of embryo sacs, and second one is Enothera. But in Enoth type, one meiosis but only two mitosis. Generally, in monosporic type of embryo sacs, one meiosis, three mitosis. But exception is there in Enothera type of monosporic, only one meiosis. One meiosis is common, but here only two mitosis will occur. Here three mitosis, but here two mitosis. We will discuss how. So in monosporic, we have two types, in which we have two types. So they are first polygonum type, and second one is Enothera. We will discuss each type in detail, polygonum type and Enothera type, how they involve in formation of embryo sac. Yeah, here first polygonum type. Yeah. It is the most common type in angiosperm. Most common type. How means 70% of the embryo sac will form by this polygonum type of method in angiosperm. Most common type of formation of embryo sacs by polygonum type 1. In which polygonum type from one functional megaspore from chalaja end. Very important. Here already we discussed in monosporic only one functional megaspores are involved, and these functional megaspore from chalaja end, and in which polygonum type one meiosis and three mitosis will occur. How means we'll discuss here generally here. Yeah. So it is a new cellus cells. Think it is a new cellus. We discuss new cellus about in ovule 2n, and there are thousands of nucellus cells are present are within the are enclosed within the integuments in ovule, and among thousands of nucellus cells, any one of the nucellus cells get modified or changed into megaspore mother cell. It also has 2n, and from this megaspore mother cell, first meiosis division will occur. Generally, in meiosis there are two sequential steps: meiosis 1 and meiosis 2. And meiosis 1 and meiosis 2 together call only one meiosis. So here first meiosis occur in megaspore mother cell like this, then it form two nucleus division, and it is a meiosis 1 result. And from this meiosis 1, and this undergoes meiosis 2 division, then it form a megaspore with four four megaspore only. Like this, four megaspore will be formed by end of the meiosis. See, this meiosis 1 and this meiosis 2 together known as only one meiosis process. So with this one meiosis process is completed. By the end of the meiosis, there is a formation of four megaspore. Here four megaspore will be formed by meiosis. And then among these four megaspore, any one of the megaspore get functional, and the remaining three megaspore get degenerated. So the important point here, and here this side we have chalaja end. Here we have chalaja end, and this side we have micropilar end. And here point is only one functional megaspore from chalaja end. Means this one only functional. See, this one only functional, and this remaining towards micropilar end, these three megaspore towards micropilar end get degenerated. Very important. Get but only one megaspore, megaspore from chalaja end functional, and from these functional megaspore undergoes various mitosis division to form embryo sac. How means, for example, here we'll do another. Yeah, here same, and this here only I am drawing here. Yeah, so here one functional megaspore, and this functional megaspore which undergoes mitosis 1. By mitosis 1, there is a 2 daughter cell will form, like that 1, 2. Here, notice first nuclear division occur, then after it follow the cytokinesis, cytoplasm division. Here nuclear division is called karyokinesis, cytoplasm division is called cytokinesis. So always here first nuclear division occur, that is karyokinesis. So by karyokinesis, mitosis 1, then it follow to nucleus, and from here, and these 2 cell stage megaspores which undergoes mitosis 2, and then it form 4 nucleus stage called here 4 nuclear stage of division will be formed from these 2, and these undergoes mitosis third division, then it form 8 nucleus stages like 1, 2, 3, 4, and this side 1, 2, 3, 4, like that 8 nucleus stage. After formation of 8 nucleus, then this megaspore which undergoes cytokinesis division, means cytoplasm division will occur. By the cytoplasm division, then there is a some changes will occur like this, and these 3 cells here three nucleus here like this arrange, and 2 nucleus arrange in center, and 3 nucleus arranged towards the chalaza end. So after cytokinesis, and around this nucleus there is a formation of cytoplasm like this, and here in central only one cell with 2 nucleus, and here same like this. So like this arrangement will be occur after cytokinesis. If you observe in this case, so here towards chalaza end we have 3 cells, these 3 cells together called anti cells, and at center we have only one cell but 2 nucleus called central cell. This is what central cell. And at the towards micropylar end, here we have egg apparatus, and these egg apparatus which contain one egg cell, middle one. See this one egg cell, and in the side of these egg cells we have 2 synergids. 2 cells are synergies. 2 synergies, one egg cells together called egg apparatus. So this is how the embryo sacs which develop from the a single functional megaspores. So this type of development is most common type of development of embryo sac from single functional megaspore. The type is called polygonum type. So likewise, the embryo sac will be formed in this polygonum type. And we will discuss another type of monosporic, that is Enothera type. Yeah, here we will discuss Enothera type. And I forgot in polygonum type there are 7 cells and 8 nucleus are present. And presence of these 7 cells and 8 nucleus is a characteristic feature of angiosperm embryo sacs. It is a characteristic feature of angiosperm embryo sac. So here embryo sac characteristic feature is presence of 7 cells and 8 nucleus. Yeah. So now we move on another type of monosporic. Sorry. Yeah. Here second one, that is Enothera type. So this Enothera type, which is rarely found, and generally it is found in evening primrose plant. And this evening primrose plant also called as Enothera lamarckina plant. Enothera lamarckina. Yeah. There is a scientist, we know that father of mutation, called Hugo de Vries. He worked mutation on this plant only. Hence, generally he was considered as a father of mutation. He worked on this Enothera lamarckina plant for mutation theory. Yeah. So in this Enothera type, how embryo sac will form, we will discuss here. In which also one functional megaspore involved, and here in which very important, in which one meiosis and very important only 2 mitosis. How means we'll discuss now. Here same as it is. Here new cellus cells, new cellus 2n. Any one of the new cellus cells undergoes modification to become megaspore mother cell. It is also 2n, and in this megaspore mother cells it undergoes meiosis 1 to form 2 nucleus cell stage megaspore, and this undergoes meiosis 2, then it forms 4 nucleus stages of megaspore mother cell, and then it follow the cytokinesis, then it form the 4 megaspore by meiosis 2, and this meiosis 1 and meiosis 2 together only one meiosis only, because meiosis occur in 2 stages, meiosis 1 and meiosis 2. By the end of the meiosis, there is a formation of 4 megaspore. Yeah. Among these 4 megaspore, only one megaspore is functional. Yeah. And here, which megaspore get functional means that is the important. In polygonum type, chalaja and megaspore get functional. But here, towards micropylar end, see, this is the towards micropylar end, this is functional. This one functional megaspore. This is functional megaspore towards micropylar end. And this rest of these 3 megaspore get degenerated. So here the degenerated megaspore towards chalaja end. So this is the important point. So in Inothera type, the functional megaspore from micropylar end. Yeah. After, and this functional megaspore, see here I am writing. See, wait. Yeah. And these functional megaspore here. This functional megaspore which undergoes mitosis 1, then it forms 2 nucleus stage of megaspore, and then it undergoes mitosis 2, then it forms 4 nucleus stage of megaspore. And after karyokinesis, then it follows the cytokinesis. Before cytokinesis, these 4 nucleus get arranged like this, and here 1, 2, 3, 1 is center, and then it follow the cytokinesis. By cytokinesis, around this nucleus, one cytoplasm will occur, means one cell will be formed here, and here 1, 2, and 3 after cytokinesis. So by the end of this, then here we have seen only one central cell. Very important, one central cell, and it contains only one nucleus. But here towards micropylar side or micropylar end, there are 3 cells are there, and these 3 cells together called egg apparatus. Among egg apparatus, the middle cell is called egg cell, and the side cells are called cyner. So likewise, in Inothera type, there are only 4 cells are formed. One cell is central cell, and 3 cells are egg apparatus. In egg apparatus, one cell is egg cell and 2 cells are synergy cells. It means that there are 3 plus 1 cell will be formed. Here 3 cell is egg apparatus and 1 cell is central cell. But in polygonum type, there is the arrangement of cells are like this. How means 3 plus 1 plus 3. So total 7. Here 3 is towards micropylar, then it is egg apparatus. Here 1 cell is central cell and 3 cell is antipodal cells. But in Inothera, only 3 plus 1. Only 4 cells are formed in this method. So this is all about the monosporic type of sac. Now we move on another type, that is bisporic type of embryo sac. Here second type, yeah, bi type of embryo sac. So here in which embryo sac form from 2 functional mega, and in which we have 2 types. Here also we have 2 types. They are allium type and one more is endymion. And here allium type in which from 2 megaspore from chalaja end, very important, 2 megaspore from chalaja end, and here in endymion type 2 megaspore from micro end. And here important note, in bisporic type of embryo sacs, in bisporic type of embryo sacs, 1 meiosis and 2 mitosis will occur. Now we will discuss how this allium type and endymion type will produce female gametophyte, that is embryo sacs. We will discuss first allium type. Yeah, here about allium type. So here mother cells like 2n, and this megaspore mother cell undergoes meiosis 1, produce same, and then it undergoes meiosis 2, produce 4 megaspore. So among 4 in allium type, only 2 functional megaspore, that is towards chalaja end. For example, here like this. Yeah, here these 2 functional mega from chalaja and will participate in formation of embryo sac, and these 2 are degenerated. Yeah. So here and these 2 functional megaspore, so which undergoes mitosis 1, then it form from here, from one megaspore there is a formation of 2, and from another megaspore there is a formation of another 2, and this will undergoes mitosis 2, then it form 4, 1, 2, 3, 4, here 1, 2, 3, 4, and here 8 nucleus stage of megaspore will be formed by 2 mitosis only. So after that, and this karyokinesis followed by the cytokinesis, before like this arrange 1, 2, 3, 2, 1, 2, 3, like this arrangement of nucleus will occur, then it follows the cyto like this, and there is a formation of cells, egg cells, synergid cells, and central cells, and antipodal cells. See, so this is towards micro parent. This is chala giant here, chala giant. So here towards micropellarent, we have three egg apparatus cells. Among egg apparatus, the middle cell is called egg cells are ovum, and these two cells are called synergids. This is what central cell contain two nucleus, pronucleus, and these three cells together called antipodal cells. Okay. So here the formation of here how this is how embryo sac will be formed by the allium type. So in this allium type of method, so here the formation of cell also like 3 plus 1 plus 3 only, like polygonum type in monosporic. Here first three cells are egg apparatus cells, one cell is central cell, and three cells are antipodal cell. It is like polygonum type. Now coming to same endermion type, here I am writing, yeah, second one, endermion type of bisporic, endermion type of, in endermion type same, the division of megaspore is same, but here we have some differences, like in endermion type two functional megaspore from micropylarent. There is a only one differences between allium type and endermion type from micropylarent, and the remaining division of megaspore into embryo sacs as it is. Like here also same, like megaspore mother cell will undergoes meiosis 1 and meiosis 2, then it form 4 megaspore. Among 4 from these two, from towards micropylarent, these two are functional and these two are degenerated, and these two megaspore undergoes mitosis 1 to produce 4 nucleus cell stage, and this undergoes mitosis 2, then it form like same 8 nucleus stage, and then after it arrange into different locations, and then it become, then after karyokinesis cytokinesis will occur, then it form same as it is 3 plus 1 plus 3 cell stage. Same, the division of allium type and endermion type is same, and the formation of embryo sacs in 3 is to 1, 3 by 3 plus 1 plus 3 only, where the major differences between allium type and endermion type is only, that is functional megaspore from which side. If two functional megaspore from allium type, two functional megaspore from chalagiant, then it is allium type. Two functional megaspore from micropylarent, then it is endermion type. That's it. So this is all about the bisporic type of embryo sac. Now we move on last and most important, that is tetrasporic type of embryo sac. Here third one, tetra type of embryo sac. Yes. So here in this tetrasporic type of embryo sacs, here all 4 megaspore get involved in formation of embryo sac. It means that there was no degenerative megaspores. And this tetrasporic type of embryo sac, also known as Edoxa or plumbago, are also called performia. Pepro. So all these Edoxia, plumbago, and pepromia all are another name for tetrasporic type of embryo sacs. Tetra type of embryo sac. So here in which tetrasporic type of embryo sacs only one meiosis and only one mitosis will occur. How means we'll discuss in last slides. Yeah, here. So this is what megaspore mother cell which undergoes meiosis 1, it form 2 nucleus stage of megaspore mother cell, then it undergoes meiosis 2, then it form 4 nucleus stage, then it follow the cytokinesis, then it form 4 functional megaspore, because meiosis 1 and meiosis 2 together only one meiosis, and from this 4 megaspore, all these 4 megaspore are functional. So hence from this 4 here from these 4 functional megaspore, all these 4 functional megaspore will undergo only one mitosis, then it form 8 one from here, one nucleus to one from 1 to 2 nucleus will form like that. 8 nucleus stage will be formed in megaspore, and this 8 nucleus stage of megaspore, then it undergoes cytokinesis division, then it become like this. Here first nucleus arrangement will occur, then after it follow the cytodivision like this. So around here, this is what central cell in sorry, not central cell, sorry, it is a egg cell, and synergid cells, and here central cells, here antipodal cells, like this will be arranged. So if we mention their name here, see here, so this region is micropylar region. Here we have egg apparatus cells, egg apparatus, in egg apparatus we have one egg cell and two synergids. And here central cell in which one cell, but it has two nucleus. And here towards chalaja end, sorry, towards chalaja end we have three cells. These three cells together called antipodal cells. So this is the how this embryo sacs will form by from 4 mega functional megaspore in tetrasporic type of method. So this is all about the different types of formation of embryo sacs in angiosperms. So with this, we have completed each and every concept regarding to this embryo sacs. Now we have few MCQs from this topic. Yeah. So this is the first MCQs here, and I forgot one thing. And in tetrasporic also, in tetrasporic type also here. Sorry. Yeah. In tetrasporic also there are formation of embryo sacs will be same 3 plus here 1 plus 3 in cell. If we consider their nucleus means how 3 plus 2 central nucleus and 3. We can write if in exam if they ask arrangement of cells means you can write 3 plus 1 plus 3. If in exam they will ask arrangement of nucleus means then you can write 3 plus 2 plus 3. So see the difference, because in embryo sacs we have only 7 cells and 8 nucleus. Yeah, okay. So this is the first question, and consider the following statement regarding the development of typical angiosperm. Read these 4 and answer it. And one more MCQs. Yeah. Yes, this one. Identify the correct statement regarding the cellular organization of mature 7 cell and 8 nucleate embryo cells. And one more last. Yeah. So evaluate the statement concerning the variation and types of embryo sacs in flowering plant. Read these 4 and which of the statement given above are correct. So all these are multi-statement questions. All are based on NCERT only. So thank you. Have a nice day.